as well. And so hopefully some of the things I'll talk about tonight, make sense to you. I'm a physician as well as a scientist. So what I've been working on for the last 20 years, almost single-mindedly, is in front of you on the screen. When I was at MD Anderson in Houston as an clinical investigator, I became somewhat obsessed with why prostate cancer had this very unusual behavior of spreading almost exclusively to bone. And so that problem struck me as what we call an experiment of nature. Nature throws up patterns that demand explanation. And this was the one that appeals to my imagination and interest. And I found an opportunity to work on this problem here at Tufts Medical Center. So what you see on the right of your screen is what is a cartoon depiction of an antibody. And that is a therapeutic antibody that is designed to treat men with prostate cancer. Our research is at an advanced stage, I would say, in terms of its preclinical phase, meaning from the laboratory poised to go into the clinic. We think we have a good version of this antibody that we want to take in front of the FDA to get their feedback to see how we can get it into clinical trials. So it's more than just a gleam in my eye or our eye. It's something that's very real and tangible. Taking an idea from a problem, an experiment of nature, of the bone metastasis, and coming up with an idea that you can target with an antibody is, has been a labor of love, I would say. And obviously the hope is not realized as yet. It has to go into the clinic. It has to be safe. It has to be well tolerated. And obviously it has to work. So all of that remains to be proven. But that is the takeaway message from today that I'm giving you right up front. And what's different about this antibody, and the title reflects it, it's reprogramming prostate cancer. It's not actually killing any cancer cell by itself. What it's doing is changing its essential nature. It's rewiring the cancer into a form that is now visible to the immune system, whereas it was not prior to that. And having rendered it visible to the immune system, it is now vulnerable to attack by the host's own immune cells and can be eliminated. That is what reprogramming, the intent of the reprogramming is. So on the left is the by the way, do you see my cursor moving as a pointer?
Yes,
John?
we can see that.
Okay. So he has a prostate, sitting under the bladder, where, it functions essentially to liquefy semen. That's his main job. But as we get older, it comes in large, causes benign enlargement, also cancer. These are the two, and inflammation. These are the major pathologies of the prostate. And as I said here, this is a bone scan showing bone metastasis, a rather graphic example of one, not intended to be frightening, but it's high volume bone metastasis. And as I said, in such a patient, it might be exclusively in the bone to exclusion of other organs like liver, lung, lymph nodes, even, and so forth. All right. We've advanced this bispecific antibody to the US Patent Office. There's actually a patent that my employer by rights holds, but obviously anybody who looks to develop that would have to license it and move it forward. I just want to give tribute to your colleague. I learned you've just lost one of your beloved colleagues who was instrumental in getting the tribe started years and years ago, and he fought the good fight. And his life is everything that we are working towards. A lot of men who die of prostate cancer do so, I think, in their prime. They may be grandfathers, but they have a life of wisdom and experience, a lot to share. Of course, I have lost young men, most recently 41 years old, with young children at home. So it does happen even in young age. And they have a different biology that's very akin to the one that we are actually targeting. So my condolences to the group, those who are present and those who are not, for the loss of Tracy Morgan. But this fine-looking man here is the one who introduced me. He was my patient, as he said, maybe 12 years ago. And he asked me to talk about it again, which I'm happy to. So he presented in the age of 59 with what I call locally advanced high-grade prostate cancer, which the prostate was just full of tumor. We can examine it by digital exam. So bulky nodular disease. Whenever I see that in a young man, I'm always worried that this conceals high-grade disease of the type to evolve to what is called neuroendocrine carcinoma. And I don't like sending men with this type of phenotype, especially the young, to radiation and hormones. I don't think it works very well. I always think to myself, how do you get this surgically under control? How do you combine systemic therapy with that? And then after it's removed surgically, consider radiations like triple therapy approach. But as it turned out, the cancer just flattered to deceive in terms of its response to hormone therapy, which was only brief. And as I suspected it would, it showed progression very early on hormone therapy. We gave chemotherapy and removed the gland as soon as we could. But then it came right back after surgery, looking like neuroendocrine carcinoma under the microscope. I started on second-line chemotherapy. That's never a good thing if you're already on your second line of chemotherapy within the first year of your diagnosis, certainly with a life-threatening phenotype. So this was a nasty clinical problem. And one that one never likes to see right away. But right across the river here in Boston was a new institute called Foundation Medicine, founded by two scientists from the Broad Institute in Cambridge, Levi Garraway and Matt Marson, who developed a new technique to rapidly profile tumors for their mutations. So we know that in cancer, genes undergo these shifts in their code of mutations that endow them with cancer-promoting properties. And what Levi Garraway and Matt Marson thought was that if you could profile individual men's tumors, we could come up with individual solutions based upon the gene mutation profile, and we could find a way to do it very rapidly. So this was transformed into a business proposition, the Foundation Medicine, for really one of the leaders in the field and remain that, although there are others certainly. And this is the type of string of mutations. There's a whole list, by the way, left to right, of mutations of prostate cancer. They can be thrown up by such an approach. You can see right here, point to a P53. It's like the guardian of the genome, very commonly mutated in a host of other cancers. And you can see this, what we call the long tail of mutations, each of these individual genes diminishing frequency. And here is a list on another, rendered into a table form in terms of classes. And right here with the green arrow was the mutation of a gene called polymerase, POLE, or DNA polymerase eta. You can see how rare that is, only 0.1%. But this was the mutation in John's tumor. And what is it that made it special? Well, DNA polymerase eta is a DNA proofreading enzyme. Just imagine a copy editor in a newspaper is checking for errors in the script. Copy editor has to identify them and correct them. In the same way, our own DNA requires a copy editor as it replicates itself. And we are endowed with these proofreading enzymes that are supposed to do the job. But if that proofreading enzyme is mutated and ineffective, the cancer starts to accumulate lots of mutations, like these errors in the script here. And that could make the tumor particularly sensitive to immunotherapy. And here's a long list of mutations in that tumor. And buried amongst that list was this DNA polymerase eta. But we were able to collaborate with Foundation Medicine and assign this entire signature of mutations to that one mutation. That's a special technique to do that. And so the idea was, well, let's treat this with immunotherapy with a new class of drugs that now you all may have heard of called immune checkpoint inhibitors. And you have Jim Allison, a former colleague at MD Anderson. He moved to MD Anderson, and Tasuku Honjo, who discovered these immune checkpoints. What are immune checkpoints? Checkpoints are inbuilt safety valves in our immune system designed to slow down the immune response. We obviously need something to activate it, but everything needs a break to shut it down. And otherwise, if it's always on, we get autoimmune disease. They found out that these checkpoints cause breaks in the immune response. But they had a simple idea: is by blocking a checkpoint, can you treat cancer so that the immune system is turned on? Very simple idea, but that's actually worked out in a large number of cancers. And so this was the idea that we had. How about treating this with an immune checkpoint inhibitor? It was not approved in prostate cancer at that time for high mutation burdens or elsewhere, but we were able to get it. It was denied by the insurance company, but we were able to get it from Merck, which was a company that makes Keytruda, which everybody has heard of now. And so, yeah. So just to give you an idea of this checkpoint inhibitor idea again, this is the T cell. This is the T cell receptor recognizing an antigen. And here's a brake system with that PD-1, this specific example of the brake, or the checkpoint that Honjo discovered in Japan, and its ligand. And by using an antibody to either the receptor or ligand, you can block this checkpoint and activate the T cell. And in John's case, we used a PD-1 antibody, Keytruda. And the tumor, beautifully responded. We were quite delighted to see that. The PSA became undetectable rather quickly. We stopped the castration therapy, or the hormone therapy, I should call it. His testosterone came back up to normal. The PSA has remained undetectable for over a decade. There's no sign of the disease. It's obviously an exceptional outcome. Very, very fortunate and blessed, you could say, to have that outcome. But is this common? Certainly not. I pointed out that this was a very, very rare, mutation. There's another one called mismatch repair class of mutation that are also sensitive to immune checkpoint inhibitors. But you know what? It's rare to see these complete remissions that last long, even in that. You can get responses, but they don't generally result in complete remissions, and they generally don't last for, like a decade. So obviously now we think, is this cancer cured? We, hope that it is, but one never knows what nature can throw up. So yeah, we'll keep watching. But the point I want to make here, I come back to that obsession I told you about. When you see 15, 20, 30, 40 things here in a row, surely they all can't explain one thing. I just didn't think that it was logical that all of these genes could somehow explain this one phenotype. So I just felt like the mutational approach, the mutational lens through which we saw cancer and analyzed it, couldn't possibly explain how it behaved fully. And I continue to feel that way. And I'll try and explain my thinking on that as this goes on. So what are the hallmarks of aggressive prostate cancer? We all know that there's types of prostate cancer that don't spread, the Gleason 3+3 grade group 1 cancers under the microscope that are best left alone. And then there are the higher grade cancers that spread and cause problems by, threatening life. How does it threaten life? It spreads outside of the prostate. It resists treatment, including hormone therapy. It evades the immune system. And as I said, even though there's a diversity of mutations, they all funnel into these major hallmarks of metastasis, principally bone, treatment resistance, and immune evasion. So our thoughts were, well, can we find a unifying a framework for explaining that, one in which we can decode a novel way to treat the cancer beyond the standard paradigm of hormones, so and hormones and all the toll it takes from someone's body with, putting on weight and losing your muscle mass and thinning out your bones and losing your sex drive and everything, all the other indignities that treatment of prostate cancer causes to men. So now I want to take a breath and try and introduce a new idea to you. And I'm going to go back to something we all learn as medical students in first year of medicine in anatomy, and we look under each individual organ in a normal human under the microscope. We understand how things work based on their organizational principles. And I'm going to show you here, I am showing you here a normal prostate gland. And on the left is an arrow pointing to a brown layer on the outside or the base of the gland. It's called the basal layer, just basal, right? And then on the inside is the luminal layer for lumen. Here's a lumen, the empty space. And what the luminal cells are doing is secreting into content into the lumen, and that goes into the ejaculatory duct. And what the basal layer is doing is generating the luminal layer because it acts as the anchorage to something called a thin basement membrane, and it serves as what we call the stem cells, the mothership, if you will, of the gland. So each of these glands have their own basal layer here decorated in brown, as you can see with the brown hair. That's the basal layer. So here on the right is a cartoon, the basal layer in this purple, and then the luminal layer in this shade of brown. And then you have these rare neuroendocrine cells that have nothing to do with neuroendocrine carcinoma, by the way. They're just not related. So basal luminal, just think of these two layers, all right, in your mind. And keep that in mind as we go forward for the rest of the talk. Because I'm going to propose to you that the cancer is actually behaving like these layers. And I'll show you here. It's a little complicated slide. But what is this trying to do? A couple of scientists, Owen Whitty at UCLA and Dean Tang, who was at MD Anderson, actually a former colleague there, who's now at University of Rochester. They separated these two layers. They could actually did a beautiful study where they separated the basal layer and the luminal layer and looked at the gene expression profiles. So, that's called, the message that the genes are sending. That's called a gene expression profiles. They get transcribed into something called messenger RNA. It gets converted to protein. And by looking at that signature of what the genes are being transcribed into, we call that the gene expression profile. And so here's a marker of basal layers. It's a big term, CD49F high. That's basically a marker of the basal layer. Notice the names of it. You don't have to understand too much what they mean, but just take a look at, just read down that list. And then look at the luminal layer marked by CD49F low. Just read down that list. You'll tell that these lists do not overlap. The names are dissimilar. Notice that for this one, I've highlighted the MYC signaling, MYC, pronounced like MYC, M-I-C-K, like the name. And here's cell adhesion, okay? These are important to future considerations. And look over here on the luminal side, immune response and androgen receptor signaling. This is where the hormones are active. Hormones are not active in the basal layer. The testosterone is active in the luminal layer, and many of us will know that hormones are the treatment for prostate cancer for decades. And on the right, you can see what these signatures rendered in two color formats. Here's the basal layer again, and here's red for one pattern of genes that are upregulated, and green for a set of genes that are downregulated. And look at how it looks on the low side. On the luminal side, they're almost flipped, aren't they? So they're very different. This is basal and that's luminal. The signatures are very different. But what will surprise you is when you look at the bottom over here, look at what we are reading out over here. Each of these individual rows represents an individual patient sample. It refers to the fact that whether it's benign or cancer, if you organize it by the marker, it turns out to be, they look exactly the same. So a basal cancer looks just like the basal layer. A luminal cancer looks just like the normal luminal layer. Let me just flip that on its side so you can read down on this side. You can see benign cancer, benign cancer. So all these cancers that are defined by basal look just like the benign basal. All right? So in other words, the way our gland is created from the first part is how the cancers have that memory. They have that overarching memory of where they arose from. The cancers can be divided into basal and luminal types, and they look like the normal basal and luminal gland, but they're very different from each other. Is that clear? Or is that confusing? Any questions at this point? This is a new idea, I think. I doubt very much that you may have heard this before, but I do want to by the end of this talk, convince you that this is a very important and crucial organizational structure to the cancer that is relevant to this antibody that I'm going to tell you about, that I am telling you about. So guess what happens with basal? So the basal type cancers, the signatures, they start to get more and more prominent the higher you go in the grade of the cancer when you become metastatic disease or when you start resisting hormone therapy. There's a progressive enrichment of the basal signature. Remember what I said about the basal layer sitting on the base on the outside and serving as the mothership. It forms a normal function as a stem cell, meaning the mothership for the normal luminal cells. So in the same way, it is functioning as the mothership for the more advanced cancers that resist hormone therapy. That's the takeaway message. And what if we split up all of that? Remember what I said about MYC, right? I showed you that MYC target on the basal side. And here's look down here. These are cancers from hundreds of men, over 600 men, classified by basal and luminal. And here you see that MYC signature right up there, MYC1 and MYC2. So there's a very different signature for the basal cancers from the luminal cancers. We've already covered that, but I'm just showing it to you again in another from a large clinical sample of men with metastatic castration-resistant prostate cancer. And you can see that the basal type is very important. It accounts for over half, right? Yeah. And what is MYC? What's important about MYC? It's a transcription factor. What it does is, what is a transcription factor? Something that controls genes of all kinds. So it controls a whole range of genes, and it has been implicated in over 70% of all human cancers. And what I'm proposing to you is that it is very important in prostate cancer as well. The problem with MYC is that we still don't have in the clinic a way to control MYC cancers with any type of treatment. All right, so this is where I want to end up finally at the end of the talk. I'm not done yet, but I want to give you where I'm headed, all right? I want to give you, because I spent a lot of time trying to tell you about basal and luminal and how they're related to each other. How are they related? The basal layer of the normal gland will differentiate into the luminal type of the normal gland. But I've also told you that cancers behave the same way, that basal type cancers can generate luminal type cancers. And we subject luminal type cancers to hormone treatment. Guess what happens under therapeutic pressure? They drift back to the basal type, okay? So this drift between one entity and the other is called plasticity. Things are shape-shifting forms of cancer, right? Cancers are mixed. They're not one or the other. They have mixtures of basal and luminal, and these exist in some kind of uneasy balance. And the hormone therapy, however, will drive it towards basal because it doesn't cure metastatic disease. It only controls it for a period of time. And over time, the basal type will start to emerge. So what is it about the luminal type? Well, here, these cancers tend to like to touch each other, the tumor cells, and they're controlled by the androgen receptor and androgen signaling, which is where all the hormone therapy comes from, which works very well. It causes regression of tumor, but eventually in metastatic disease, castration resistance emerges. Meaning the cancer is now growing despite low testosterone levels, no matter how potent the treatment is to lower the testosterone or to block its effect. That's called castration resistance. Whereas the basal type tumors are different. They're not really as interested in touching each other as they are in contacting the environment around them. They're called tumor stroma interactions. And what I'm going to tell you is that they're the group of proteins called integrins that mediate these interactions that are crucial to the identity of the basal type cancers. What did I say again? I said the basal type cancers love to interact with the environment. They prefer to do so, and they do so with these little fingers that attach to the matrix and hold on tight, and that's called integrins. And the force that they exert on the microenvironment is what delivers the signature of MYC. That's what I'm going to tell you. And that's how MYC is coming from. It's from this interaction with the environment. Recall the basal layer of the normal gland. It's sitting on the condensed matrix of the environment. That is actually where it's remembering its identity. And this antibody is going to turn that off and switch it off so that it will force the cancer toward a luminal type and allow it to be seen by the immune system in order to allow the immune system to eliminate it. That's the take-home message of the translational research that we have developed here at Tufts. This is just published in the October issue of Molecular Cancer Research. It's an American Association of Cancer Research journal. And it was selected as the editor's pick for the month of October. So those of you who are interested in the science of it and feel like, oh yeah, I've got enough biology in my high school or college, I can digest it, I'm happy to send you a copy. This is the simplified version of the organization. Basal, luminal. Luminal is hormones. Basal is integrins. And controlling integrins is what we need to do. That's our proposition. Obviously, if you combine both classes of treatment, you might do even better, but that's for another day. All right, so metastatic disease. We think that integrins are unifying everything about these hallmarks that I referred to before. Our research indicates it mediates. That's in fact where our research started. I'm not going into all the details of how we derived this insight, but that would take too much time and too complex for this talk. But we implicated integrins in this process. Integrins, meaning on the prostate cancer cells themselves, are allowing it to home to the microenvironment and also to resist hormone therapy through enabling the upregulation of MYC. And MYC is a master regulator of immune evasion. MYC shuts down the immune response very powerfully in tumors and is referred to as a master regulator of immune evasion. So by turning MYC off, we can now render the cancers visible to the immune system. And so here's this bispecific antibody, and what is it doing? It's attacking two different integrins, one called alpha V and the other called alpha 5 beta 1. It's got a complex structure. It's what we call a tetravalent, 4 different attacking ends to it. And it's called a tetravalent bispecific. Why is bispecific? Because it's targeting 2 different targets. So back to integrins again. What are integrins, you might be wondering? I mentioned those fingers sticking out of the cancer cell and holding onto the matrix. And here are those fingers. Here's an integrin. It's got an alpha and a beta. You could call them siblings that need each other to contact this matrix proteins, and then they connect across the cell membrane, and then they connect to all kinds of complex pathways that we don't need to go into, but they endow cancer cells with a host of advantages, okay? Moving, spreading, invading, stem cell behaviors, forming new blood vessels, evading the immune system, as I said. And here's a family of integrins. There are a whole bunch of them. But what we have identified specifically is that there are specifically 2 of them, alpha 5 and alpha V, on this left side. It's not in this quadrant, but in this one here. It's alpha V with all of its funny partners, beta 3, they have these partners, beta partners, and alpha 5, which has its obligate partner, beta 1. These 2 working in concert. That is our insight, that if you block one of them alone, the other one will compensate for the other. This is a very crucial insight that our research has provided, that you have to block both of them, and not only block them both, but do so using a bispecific antibody, which actually does much better than 2 different antibodies combined together. A bispecific antibody does better than 2 different antibodies put together. That's the point of the message here. right? Yeah, that's the bispecific. So let me give you an example of exactly that. This is something called a Western blot. We're treating cancer cells with our bispecific antibody. And here's bispecific antibody treating a cancer cell in this lane here. Look what happens to the integrins. They're disappearing. Can you see that? This is integrin alpha 5, one of the ones I mentioned. This is integrin alpha V. Can you see that they're disappearing with the bispecific antibody alone in this lane with the arrows? Whereas if you use individual antibodies, whether it's alpha 5 or alpha V alone or the both combined, no such thing happens. So the bispecific is uniquely endowed with the property to induce the degradation of these target integrins, and that's shown on the flow cytometry on the right, which you can ignore. But the point of the matter is it has a unique mechanism of action. So here's that matrix thing. I want to tell you something about force. I might probably bet you there's at least one engineer in the audience, probably more than one. And what these things do is sort of act as of levers. They attach to the micro. Here's the integrins again. Okay, here's the alpha beta partner strung along on the outside. On the outside of the cell. This is the cell. We're looking cross section, and here's the matrix on the outside. Here's a matrix protein called fibronectin that the integrin touches and connects to, and it holds on to it, all right? And then by holding on to it and pulling on it, it transmits its force into the cell through this complex assembly of connecting proteins into something called the actin cytoskeleton. Now, we have our own skeleton, right? We have our bony skeleton, but our cells themselves have a skeleton made up of a protein called actin. And that skeleton is connected to the integrins, which is now connected to the environment. So you can imagine that connectivity that occurs across basal cells and the environment, or basal type tumor cells. And that force is crucial. And I told you I'll try to tell you something new about prostate cancer. Hopefully this is something new. So here's an example of what happened. Why does force matter? Force matters a lot more than one imagines in a medicine. Here's a cell on the top here that is placed in a stiff environment. And when placed in a stiff environment, it turns into a bone-forming cell. The same cell, when it's placed in a soft, I'm very simplifying this, the same cell, look at its shape when it's put in a soft environment. It's more rounded, not as spread out. And what does it become instead? A fat cell. So under one set of conditions, the same cell will become a bone cell in a stiff environment, a stiff matrix on the outside, or it's a soft matrix, it becomes a fat cell. So that's just a very simple, it's a complex diagram, but I've simplified the explanation in order to convey the idea that force matters, because when you put it in a soft environment, it can't really get that traction force to activate this protein called YAP, which is really one of the most important mechanotransducers. Mechanotransducer is something that transmits force. And as it will turn out, YAP is the crucial link to MYC. Remember what I said about MYC? MYC is a hallmark of basal type cancer cells, prostate cancer cells, that's implicated in metastasis, in treatment resistance, and immune evasion. I'm going to tell you that YAP is the link to that, between the integrin to the YAP to the MYC. All right, a lot of terminology. I hope that's not too much. And here's that bispecific, the special action of the bispecific antibody is turning off a range of proteins there, which you do not need to know all of the names, but just one of them suffices, YAP. You can see that with the bispecific antibody alone in the nucleus of a cancer cell, YAP is turned off, all right? Whereas it's not with the individual antibodies. This is crucial because this is the link to a new class of treatment for prostate cancer in our view. And what happens when you put this antibody and treat cancer cells? You can see these basal type cancer cells, DU145 and PC3. Here are the treated cells here in green. On the left, you can see that they just completely knocks out millions of cancer cells being injected into mice. They cannot grow if they're treated with the antibody. Or this is another one, PC3, even in very low doses, it knocks out the growth of these tumor cells. And when you compare it against these individual monospecific antibodies, the one that attack only one of them alone, you can see the clear superiority of the bispecific. This is a survival curve. Mice live much longer when they're treated with the bispecific compared to the other ones. Moreover, if you establish tumors in these mice, up to 50% of them will disappear completely just with the bispecific alone, whereas the other ones simply do not have that kind of action. So clearly the bispecific is superior, not just inside a petri dish in terms of controlling these mechanisms that I just showed you, but also inside a mouse model of cancer, significantly superior to the individual antibodies. A point about that, these individual antibodies, which I'm showing you here, alpha-5 beta-1 and alpha-V, were combined into this bispecific, right? And these antibodies have been taken to clinical trials before in men with prostate cancer and have shown only very little activity. But we are saying, look, the point is you had to block both of them and not just one of them alone because they compensate for each other. Not only that, if you do a combination, it just simply doesn't work as well as the bispecific. The bispecific is special. So you may have heard of bispecific antibodies. There's a huge development of bispecific antibodies in medicine, and prostate cancer in particular, relating to a class of bispecific antibodies called T-cell engagers. They're bringing T cells, immune cells, close to cancer, and then causing, and some of them are actually working interestingly well. This is the so-called STEAP1 antibody that is in clinical trials in prostate cancer, showing activity even in advanced treatment-resistant cancers. But we are taking a different approach. We're not dragging a T cell close to the tumor. We're simply knocking out the integrins and reprogramming. So what happens to tumors when they're treated with the bispecific antibody? Just look over here from left to right, and you can see this one has a lot more, looks more fuzzy and grainy. That's because a whole bunch of immune cells have just flooded into these tumors. I have a special technique here on the left that shows us how many monocytes and macrophages and what we call dendritic cells and what we call natural killer cells, they're all flooding into this tumor that has been reprogrammed by the bispecific tumor. And that was by the bispecific antibody, and that's the whole idea. You reprogrammed it and made it visible to the immune system, and now the immune cells are just flooding into the tumor. And what is causing the tumors to die? Here's the bispecific antibody in the mice. What you're seeing here on the left here are tumors growing in mice, right? This is untreated. These are treated with the bispecific. You can see there are no tumors there at all. But if you knock out the natural killer cell, if you knock it out with an antibody, you can completely reverse the action of the bispecific antibody in two different models. What does that mean? It means that there's a specific type of immune cell that is required to kill these tumors that is being activated by the action of the bispecific antibody. So we have specifically been able to implicate these natural killer cells as crucial to the killing of these cells. And this is the first time this has been shown to our knowledge in prostate cancer. Back to our favorite basal luminal idea, but I want to make the point about the reprogramming. This is a gene sequence analysis of expression analysis showing what's happening in these basal tumor cells. Remember what I said about MYC? When you see this red going to the left, that means MYC is being downregulated. This means it shows you this is the most important gene being downregulated with the bispecific antibody is the MYC target. So I've tried to impress upon you that MYC is so important to control the cancer. And here we have this bispecific controlling MYC. And that's the number one. What is the number one upregulated? That's green, and going to the right is what is going up. That's interferon response pathways. So we're controlling MYC, it's coming down, and what's going up? The immune pathways. So what are we doing effectively? Look on the right, this is what I showed you earlier. Remember what I said about MYC being top of the list in the basal type tumors? See what's active in the luminal type tumors. It's the interferon pathways, the immune pathways. So we are shifting the basal tumors towards a luminal tumor phenotype, making it less aggressive in the process and vulnerable to the immune system. And I showed you that this figure right in the beginning, introducing you to the normal prostate gland. But what I didn't tell you was the stain for the basal was in fact the integrin itself, integrin alpha V. So the integrin alpha V is part of the normal basal. It highlights the normal basal layer of the normal gland. But in the tumors, our studies have shown how it appears in diverse and striking patterns. And in bone metastases, in almost wall-to-wall expression of the integrin alpha V in a tumor in bone. So it fits very, very well with what we have described as the organizing nature of the basal type tumors, that the integrin alpha V is implicated in that very powerfully. And when we correlate, this is a correlation of integrin alpha V and MYC in men with metastatic disease, looking at the gene expression, we can confirm indeed that the number one correlate of integrin alpha V in men with metastatic treatment-resistant prostate cancer is in fact the MYC pathway. And we've done this in multiple other data sets showing that the top ranked gene association of integrin alpha V is the MYC target. TMA 100, this refers to a rapid autopsy series, men who had died of prostate cancer and donated their bodies very generously to research. And those tissues are available for analysis. And we were able to collaborate with the University of Washington and show that principal association of integrin alpha V with MYC in such tumors, but also in other data sets of metastatic castration resistance. Remember I told you about the 41-year-old man with the young children who died of prostate cancer? This is DKFC is a data set in men who had prostate cancer below the age of 50. And in men with early onset prostate cancer, the integrin alpha V correlates with MYC, number one on that list as well. You can see the contrast within integrin alpha 5. It's very different. But right at the bottom, you can see another one called EMT that integrin alpha 5 is associated, also associated with stem cell biology. So both integrins do matter. I covered this before. So I want to try and get to the end of this talk by telling you that I've been trying to tell you about the integrins, what they are, how they connect cells to the matrix, and how they exert force on the matrix in these basal cells. And that force, you can see the tension here, is what drives the second messenger called YAP into the nucleus of the cell and triggers this MYC pathway that shuts down interferon signaling, making the tumor cold and evasive to immune system. Cold means it's not infiltrated by immune cells because it's excluded because of the action of MYC. But if we reverse that with the bispecific, blocking these integrins, shutting down that force principle, excluding YAP from the nucleus, now the MYC can no longer function because the MYC requires that YAP to set it off. And now the interferon response is unleashed, and all sorts of cool things are made by the cancer cell itself, which allow the immune cell to recognize it and come in and inflame that tumor and kill the tumor cells. And that's the general idea behind the whole concept is sort of put together in this cartoon, and I hope it makes sense from the explanations I've been giving you. So back to this idea, this is where we are, the basal luminal trajectory. Can you notice I've not said a thing about mutations, right? We started with that long tail, and I didn't say a thing about mutations. Point of the matter is mutations funnel into this structure. They don't define it, they just funnel into it. This is what defines the organization of prostate cancer in an overarching sense. And I think the mutations that we see only feed into it and enable it and in some form. So from 1941 onwards, we've been treating prostate cancer with hormone therapy. As I said to you, this is the whole thing we learn as medical students, how hormones are regulated in the body. Suffice to say is we're trying to knock out testosterone and its signaling. And this is the surgeon from the University of Chicago who first showed that testosterone was required for prostate cancer functioning. Gave us the idea of knocking out testosterone signaling. We've held on to that and developed it a lot in the decades since, but these are essentially targeting the luminal type cells, right? Remember I told you that it's the luminal cells that control that are being controlled by androgen signal, but not the basal cells. And the basal cells are the ones that are the most lethal component of prostate cancer. And so when we have unified all of the hallmarks of lethal prostate cancer under the concept of basal and integrin-mediated mechanosignaling. And we think that that whole enterprise is can be controlled by the bispecific integrin antibody, and that mutations of genes like P53 and PTEN, loss of the androgen receptor, the BRCA, even the BRCA1 and 2 that you hear of in hereditary syndrome, they all feed into this mechanism. That's our theory. There's evidence for it, and we think that it provides a more organizational logic that makes a lot more sense, because now we can all explain how it's all men are getting the same bone metastases. It's because it's the same basal integrin regulated thing that loves the bone, and these mutations just feed into it, fuel it. They don't actually define it per se. All of this work, I would say, has been done by this young man, Raghav Joshi, who is an amazing colleague I've worked with for over a decade. A wonderful temperament, wonderful character. I'm very proud to have him as my colleague. And a whole bunch of other people who came to the lab over the years who contributed in different ways. But he has been the one constant over this length of time. Very grateful to the National Cancer Institute for their support. We have a renewal of our R01 grant going in next week, in fact. So I'm giving this talk in between our preparations for that grant submission, in which we hope to show a broader application of a bispecific antibody to other malignancies, because we think this basal phenotype applies to breast cancer and ovarian, pancreatic, and other cancers. And we intend to demonstrate that in future studies to come. Speaking to this audience, I want to salute all the families with prostate cancer who find that that life has dealt them that challenge. And I have to say that none of my work would have been feasible without the support of my patients and their families. This is Julie and Will Person from Texas, who first met me at MD Anderson and supported my work. Will passed away from metastatic prostate cancer. Julie continues to live in Texas or in Denver now. And these are the Denorcias from Florida. By the way, they give me permission to share their names, so it's not an issue. Brenda and Anthony Denorcia and their young children. Losing your father at a young age is very difficult. He had neuroendocrine prostate cancer, and Brenda's mission is to find a solution for a horrible problem that robbed her of the love of her life. So they have been major supporters of our research, enabled us to get our R01 and supplemented the work that we are doing. Mike Rosenblatt, I love giving him a shout out to him. Visited MD Anderson in 2008, invited me to join him at Tufts, where I met Eddie Goiberg. We both worked together to set the foundation for the studies that Raghav took up later on and developed. And thanks to John White and the Mohawk Mission for this invite. It's been a pleasure, a privilege to speak to you. I'm certainly happy to answer questions in the time we have left. Hope I didn't take too much time.
Thank you very much, Doctor. If you can put your slideshow down, that will allow us to have more visible,
yeah.
The thing more visible. And
yep. Let me see. I don't want to shut it off completely. Let me see how I do that.
Okay, I think that's all right. It looks like as soon as someone raises their hand, they'll come up on my screen. So,
okay,
Kevin, if you want to unmute yourself, and I will, before you start, Kevin, I will say, please, gentlemen, keep our questions as concise as possible. Thanks.
Thanks, Ed, and thanks, Doctor. And first, I just want to say thank you so much for finding this to be a passion in your career. It's so specific and it's so difficult and complicated, and I'm just amazed that you spend your days doing this, and thankful that you do, because it means a lot, especially I think specifically to the neuroendocrine kind of flavor of this is relatively rare. And I've just found myself diagnosed last February with it and kind of working through it. So just a couple of quick questions, I guess. So the one you mentioned, your interest that the cancer spread to like the bones most often and not everywhere else. And I think you kind of alluded to at the end, like why do you think that is? And is there a specific aspect of the cell mutation that makes it want to go to the bones?
Yeah, I think it is very intrinsic. What I think the answer is that the integrins are playing, that I mentioned, are playing a crucial role in that affinity for the bone and the efficiency by which you could say it colonizes the space, the bone marrow space. It just gives them unique advantages. And there's a match between the prostate and the bone in ways that go beyond just that equation alone. Very likely, the calcium content in bone triggers the function of these integrins. The low pH, the low oxygen environment triggers the action and amplifies it. So it gives it a momentum and that is exceptional. It's not that the integrins cannot allow the cancer to spread to other organs. It's just that the efficiency quotient in the bone is enormous. And that's why it lends itself to that kind of efficiency. That's our current working. So we've implicated these integrins in the spread to bone, but we haven't said this is why it does so preferentially, because there are multiple other parameters that are likely feeding into it. So it's what we call multi-parametric. And to discover all of the others is difficult to do because there are no animal models of prostate cancer spread to bone. We're only dealing with very relatively crude cell lines and other things that we use in the laboratory that really are only a partial facsimile of what really happens in nature.
Yeah, you also you're talking about John's identification of the POL mutation and how rare that was. I was frantically looking at your screen there. I have an ATM mutation that has been identified back in August, and I noticed that seems to be a little bit more popular. But I've been kind of I understand that there's still not like a BRCA1 or BRCA2 where there are like active immunotherapies that could be helpful with that. Is there anything I can do to kind of like push the ATM, look at the ATM further?
Yeah, I mean, I think the ATM belongs in the class of genes that have been implicated in DNA repair, or what is called double-strand DNA repair, akin to the BRCA family. But the sensitivity, not so much to immunotherapy, but to a class of drugs called PARP inhibitors, PARP, is where the action is mostly for the BRCA or the DNA, repair mutation signature. That's the class of drug you are probably going to be talking to your oncologist about, a drug like olaparib or niraparib or talazoparib, which are the three ones that are currently available and approved in the United States. I think, for ATM tends to be less sensitive to that. So than BRCA to these class of drugs. So other strategies are being looked at, including something called the ATR inhibitors that are in clinical trials and being used in combination with the PARP inhibitors. So there may be a clinical trial that could be specifically available. I can just say that for now, I also treat my patients with ATM mutant castration-resistant prostate cancer with PARP inhibitors, like olaparib. In fact, I had a patient just today. I saw a few hours before coming to this talk. He's actually doing quite well so far on olaparib and abiraterone. But we know that that class of drug does not really excite the immune system. Mm. So, we can't, combining PARP inhibitors with immune checkpoint inhibitors, the results haven't been great. And so we definitely need a transformative approach, a transformative way of looking at these mutations, such as the BRCA and others. And the last figure I showed, which I recognized was getting complicated, was the idea that the mutations feed into that MYC-regulated paradigm, right, that is controlled by integrins. So it could be that ATM as well as BRCA and others, feed into an integrin alpha-5 and alpha-V regulated superstructure. And if you control that with the bispecific, you can now get a real bang with the immune system and eliminate them better than the PARP inhibitors. That, of course, is unproven as yet, but it's in our plans for future study in a high priority fashion.
Great. And then, sorry, I don't want to monopolize, but just one more quick question. That do you have any experiences where neuroendocrine presents but still reacts well to traditional treatments? Like that's where I am right now. That I had started at a PSA of 3,000 and I really lit up like a Christmas tree in my PET scan. I just had my oncology appointment today where I responded very well. My PSA is down to a 0.76. Even though the biopsy of the tumor presented as neuroendocrine, and that tumor's not there anymore. So it's kind of confusing about, I mean, it's good news, right? But like, have you seen that before?
I mean, I would scrutinize that a little more carefully. A PSA of 3,000, generally means a more luminal type cancer, not a basal or neuroendocrine type. Of course, there are admixtures, and so the standard of care in this situation, assuming there's widespread metastasis, as you said, would be what we call triple therapy with docetaxel, darolutamide, and, for example, Eligard. This would be sort of our approach. And then, of course, I would be very early in getting next generation sequencing of the tumor, very early in doing clinical genetics, talking to a clinical geneticist to make sure you're not harboring a BRCA gene. So that would be the approach. But I don't think the neuroendocrine emphasis has immediate relevance at this point. I think the main point is, can that PSA get down to undetectable levels below 0.76, to less than 0.1, less, if you can get to that, that would be a great goal to with triple therapy.
Great, thank you.
I know people have their hands up, so.
Brian, go ahead.
Okay, so I happen to have an MSH2 genetic fault, or Lynch. And I know my doc had mentioned something about some kind of something that is heavily within like MSH2 and other genetic faults that research has recently lent towards, immunotherapy being effective against it. But I know earlier when you were talking, you said that basically the way that immunotherapy works is it shuts down the inhibitors and excites your immune system. Well, I've also got other autoimmune stuff. Does that in effect help that, or does it kind of send it into overdrive and then make it kind of pull back? And I'm not metastatic yet. I probably will be at one point or another, but I'm very low on different versions of treatment that I will have available to me for the next time when it comes along. So the immune therapy stuff seems very promising, at least, but I didn't know if you couple that with the autoimmune, if it kind of puts it in overdrive and makes it less effective.
So you're saying you have an underlying autoimmune illness of some kind?
Yeah, I've got vitiligo, and there's another autoimmune that I also have. I can't remember what it is, but then I've got the genetic fault thing, and I've got a long history of cancer at a very young age. So,
oh, okay. Well, it does depend. Not all autoimmune illnesses forbid the use of immune checkpoint inhibitors. So I think it has to be a calculation of risk benefit. If the cancer is very minimal at this point and non-threatening, this may not be a time to consider it. But on the other hand, immunotherapy may work better when the cancer is low volume. So these are some of the tensions that have to be weighed by your oncologist. But if the autoimmune illness is severe enough, then it could be a big mistake to do an immune checkpoint inhibitor and run up into a bigger problem than the cancer. So that's always always a challenge.
I got you. Is there anything else? And I know this is outside of what you're talking about now, but like I've had two separate cancers in the same spot, and they're 10 years apart from each other. I was diagnosed with my prostate stuff at 37, and I had radiation again for a second time. And so I'm pretty much done with radiation, which, you know, with Lynch, it's all about your, you know, mostly colon, small bowel, stuff like that. So it's all in that same kind of area.
Yeah.
And I know you're a prostate guy, but you know, it all lends towards the same treatments apparently. With
Yeah.
regard to the immunotherapy side of it, is there any other things that are outside of immunotherapy that are also
Well,
outside
I mean,
normal stuff that's promising now?
well, I mentioned the BiTEs, the bispecific T cell engagers at the clinical trials, which may become relevant in your case and which may not be as threatening as an immune checkpoint inhibitor to activate your immune system, only because they mean bringing the T cells directly to the cancer cell itself. So that could be something to think about, because those trials, the STEAP1 in particular, is moving to phase 3. So that may be coming down the pike in the not too distant future. So I do think there are ways to harness the immune system in the context of someone who has a profile of autoimmunity through a technique such as a bispecific T cell engager. So that is for the future, of course, but it's something to think about.
Very good. I'm not there yet, but like I said, I know with the way my life's gone with my genetics, I will be one way or another. So
Yeah. A good partnership with an oncologist and a good cancer center, tertiary cancer center, for a complex case such as this situation, sounds like to me would be very important.
I go to KU currently, but I've also dealt with Dana-Farber.
Okay, good. In good hands then.
So and my doc at KU wouldn't hesitate to send me elsewhere if I needed to go that way. So
Sounds good.
all right, very good. Thank you.
Yep. Got a couple more hands raised.
Dave Hedrick, please. You have to unmute. You're muted, Dave.
Excuse me. Thank you very much for your work. Thank you, Dr. Joshi, as well. My question is about STEAP1. My oncologist at the University of Washington was John Lee, who's now at UCLA, and he was working on this. And I didn't know it was so far advanced that it was going to stage 3. Is that complementary to the work that you're doing? Could we expect some relief from that as we wait for the bispecific antibody?
Yeah, I think they're definitely complementary in a sense, because we are not directly activating the immune system or dragging a T cell to the proximity of a cancer cell. What that STEAP1 antibody is doing is dragging a T cell to the proximity of a cancer cell, and then it works. Lo and behold, like knocking two heads together, it actually works and seems to overcome some of the hostility that the microenvironment in which cancer cells love to thrive. It's able to penetrate through all of that fog and make it work in bone or in liver and places like that. So that's quite unusual for an immunotherapy to have that profile. And there have been many other bispecifics that simply haven't had that kind of activity, as this one seems to show with the early reports of its activity. The one that we're working on is very different. And as I said, from the beginning, we are reprogramming the essential lethal nature of basal type prostate cancer, the type that loves to spread, to evade the immune system fundamentally through activity of MYC. So yeah, we think that these two could readily combine with each other. Certainly our bispecific could combine with other things too, like hormones, obviously, or with immune checkpoint inhibitors, or you name it. I think this antibody is in a class of its own because of its potency in reprogramming the cancer cell in its very essential sort of foundational origins from the basal layer of the prostate. Of course, there are many unresolved questions. Will it be toxic? Will it cause some undesired toxicity when taken to clinical trials? So these are all the hurdles that have to be crossed.
A little follow-up. Does your model provide a space for the introduction of things that may cause reprogramming, like pioglitazone or metformin, that have been somewhat investigated?
Yeah, I think those are somewhat, as I say, distal to the whole idea of MYC action. So MYC, remember, I like talking about MYC because I think it's like the master regulator of the cancer phenotype, especially the stem cell metabolic phenotype. We talked about its immune evasion control, but it also controls metabolism, and which is what you're referring to with pioglitazone or metformin. And I think, MYC, controls either a shift to one type of metabolism called oxidative phosphorylation or to another type called the glycolytic pathway, depending on the needs and the specific identities of the tumor. So I think MYC is sort of the master regulator of the cancer metabolic phenotype, whereas metformin and pioglitazone are somewhat downstream. So I sort of like to think that we are at the source of the whole business. And that would be a much more potent proposition.
Thank you.
Dave Plunkett.
Thank you, Dr. Matthew, for that presentation. That was a lot of information. It's going to take me quite a long time to digest what I came across, but that I enjoyed that. So thank you. What is your path forward? What do you see that needs to happen next in your research, and how will that spread to patients across the nation and across the world?
No, absolutely. I mean, I think there's something called the Valley of Death that refers to the passage of a discovery such as this one from an academic laboratory such as ours into clinical trials. And that just is a filter for, many ideas that come out of universities and the funding requirements. It's extremely expensive to take a drug to clinical trial, unlike, say, bring a new app out in tech in Silicon Valley, right? It's a very different proposition. It costs something between $10 to $15 million for a first-in-human trial of an antibody, even with a small number of patients. So now we need, obviously, FDA review of an approval of a first-in-human investigator new drug application. That's one hurdle. The second hurdle is finding the funding either from pharma or venture capital or angel to fund a first-in-human trial to show what is called proof of concept, meaning it is safe and it's well tolerated in humans and therapeutic doses, and that it's showing promising signs of activity. Once it reaches the so-called proof of concept inflection point, then it becomes much more de-risked. I'm using parlance from the industry. It's de-risked for further investment from the bigger VCs or to be taken up by pharma. So that is a huge challenge. It's not our natural skill set as doctors, We're not really entrepreneurial as such in terms of the classical sense of what an entrepreneur is, although our activity certainly is in many sense entrepreneurial in terms of a problem-solving, approach to life. But that is essentially the key challenge for us. It's a funding, mostly a funding issue, and there's a giant sucking sound coming from AI funding. All the money is rushing to AI, and as you much have heard many times over. Everybody wants to make a killing in AI, so I don't think there's that the funding constraints have dropped. Never mind the cuts at the NIH, all of that. There are many environmental challenges for us to grapple with in terms of getting this antibody to the clinic. But obviously, we think we've passed enough tests with our science. Our science is very rigorous. It's been pressure tested with many key opinion leaders in the field, shall I say. And obviously our publication and our recognition as the editor's pick for October is part and parcel of that. So I think we also think our antibody is fit for purpose. In other words, it can be manufactured at scale. We've done all that sort of thing, and can be dosed every 3 weeks. So we've passed many sort of, you could say, tests that show that it has promise for testing. We just need a funding funding source to get it into trials. Yes, someone, Ed.
Yeah, I would think that most of the gentlemen in this group tonight would agree that the side effects of treatment that we endure are probably the most difficult part of this journey.
Mhm.
And I'm not sure if I missed it, or I'm curious if we were to fast forward 10 years down the road and this jumped all of its hurdles.
Yeah.
Would this be something that could avoid ADT and androgen deprivation? Removing testosterone, because that typically is one of the most difficult parts of treatment.
Yeah, I think that's certainly very much on our minds. How do we, if not eliminate it, at least curtail its use dramatically? So a scenario like this one, where you combine a short course of hormone therapy with the bispecific antibody to spark a very potent immune response, which is now capitalized upon by an immune checkpoint inhibitor to completely eliminate the tumor, particularly the basal component, which is the mothership, which will now allow us to just avoid using hormone therapy altogether. Which takes me back to John's example, right? Where we knew that the hormone therapy was simply not going to do anything for a neuroendocrine like prostate cancer. We stopped it, and the immune therapy was sufficient to control it for over a decade. So if we can reprogram cells into one that is vulnerable to the immune system, there's every reason that we can eliminate hormonal therapy if that works as well as we think it could.
We
That
will
would be-
nominate you for sainthood.
Well, that would be one thing to consider using in high-risk localized tumors, not just hormone therapy. So imagine a scenario where men don't have to undergo surgery or radiation therapy, whereas you can reprogram the tumors, if the ones that are high risk and MYC connected, into one that is now recognizable by the immune system, such that it can be treated in situ in the prostate itself. So all that you would get is an inflammatory reaction in the prostate, but not lose your prostate, not lose your sexual function, or have your urinary function impaired. So obviously that is an idealized scenario, but theoretically, there's nothing to say that it wouldn't, be testable.
Well, thank you so very much for your time this evening and for this amazing work you're doing. I don't see any other hands raised. If we don't have any more questions, Doctor, thank you. This has been, as David said, there's a lot to digest, but just what an amazing work you're doing, and thank you so much. And thank you for the work you did with our brother John, because he is such a great asset to our group.
Yeah, thank you all for the invitation. Thank you, John. And obviously it's a pleasure and a privilege to talk to you. And maybe one day I'll have something along the lines we just discussed to talk to you about, and that's the dream. All right? Take care, and I wish you all the best, and all your families as well.
Thank you. Good night.
Okay.
Thank you.